sexuality. They don't reproduce, they lack sexual reproduction. However, actually sexual reproduction contributes to which type of genetic things? It is vertical genetic transmission. However, in bacteria, other three kinds were things were found, like transmission, where the naked DNA from the environment can get in, and it can be accepted by a host bacterium, and uh, so that was shown by Griffith. Then he called it transforming factor. What is it? Of course, later it was shown that it is the genetic material. It is the DNA. Then the second, it was conjugation, wherein the bacteria, in certain bacteria, majority of them are gram-negative bacteria like E. coli. So where in bacteria, of course, in that particular chapter we have discussed, other than flagella, which contribute for motility, they do have the shorter bristle-like structures, um, which may be used for different purposes. For in case of pathogenic bacteria, for attachment to the bacterial, plant surface. The other type also were also found, that is the which are used which were used for uh, transforming the genetic material, which can occur in two forms, that is as extra chromosomal structure in the form of a plasmid, sex plasmid you call. So the one which possess that, call them F plus. The one which do not have that, F minus receives, but later to become F plus. All of them gets transformed into a thing. In other way, this particular thing, the sex factor may be, associated with the insertion sequence, transposon mediated thing, where they are actually covalently bound to main chromosome. They become part of the integral part of the host bacterium, which actually acts as donor cell. In this case, the cells which receive, of course, in the previous case, the one which receive F plus may not retain that for down the generations. They may slowly lose that particular thing. That is, you call curing. But in this case, it will remain permanently. That's why it is called as high frequency recombination. It leads to. So that's the one. Then transduction, of course, bacteriophage mediated genetic transfer. So the two kinds of bacteriophages, lytic, lysogenic, were there, and uh, the lytic bacteriophages are responsible for, of course, by chance, they may, while packing the virion particles, they may, by mistake, so they may pack a fragment of the host DNA, because which is, while lysing the bacterial cell, it is disintegrated actually, because a bacterium which is infected by a lytic bacteriophage is not allowed to multiply further. So it is permanent damage is done to the things, so that particular thing, and the bacteriophage which is carrying this information may transfer that particular character. This is a chance factor, so generalized. The next thing is the lysogenic bacteriophage, where initially the viral DNA is integrated with the the bacterial DNA, and it remains part of the post-bacterial genetic material, and as and when the bacteria multiplies, it is also carried down the generation. But at unfavorable conditions, when the virus senses that it acts like lighting, means at that time it so causes excision, means separation of the viral DNA. At that time, what may happen is a small part of the bacterial DNA adjacent to that may be carried. This particular thing is studied by the molecular biologists, and this is used in genetic engineering. Of course, you have seen that in the next subsequent things. So that's how it causes, it does that, specialized transduction. And the next thing is, of course, we have looked into uh, we have looked into then the vectors. Of course, recombinant, the principles that are involved in the genetic engineering, the vectors that are involved in genetic engineering, it is introducing foreign DNA into a bacterial cell, transgenic. You call it as transgenic technique or recombinant DNA technique or genetic engineering. It's called. Here, for this purpose, we make use of. Now, you need to carrier, molecules. You call them vectors. You call them the enzymes which are responsible for, you know, making cuts. The restriction enzymes, all that, and of course, there are various means of genetic things, but the one which are involved in plant genetic transformation, it is the agrobacterium mediated genetic transfer, which is very important. Uh, Either Ti plasmid or Ri plasmid, and which are slightly modified to remove the virulence. They think they are made binary vectors. From that they derive binary vectors, means which can multiply both in prokaryotic system and eukaryotic system. So the binary vectors. Okay. So till vectors, vectors of course, plasmids. Then further the bacteriophages. Then bacteriophages afterwards. The lambda bacteriophage based vectors are used. Then cosmids, the combination of they make use, derive certain parameters from the plasmids, certain class of parameters from the lambda phage, and to make it more vibrant. That is the cosmids, because the cosmids carry cohesive part of the lambda phage where packing becomes very easy, and larger pieces of the foreign DNA can be packaged into these cosmids. Then you have, of course, other more recent ones, like you have the bacteriophage artificial chromosome. So then bacterial artificial chromosome, sorry, then yeast artificial chromosome, human artificial chromosome. Like that, you have varieties of things. In recent years, people, the molecular biologists have constructed all that. So the next thing is a brief information on the enzymes which are useful, the genetic engineering. So you are aware the enzymes which make cuts, which cut the uh, nuclear material. They are called as, there are two types, exonucleases and endonucleases. The exonucleases, they achieve the nucleic acid strand from the external end, from the tip, Whereas the endonucleases, they make the cut in the middle. They can make a cut in the middle. So that's the advantage of them. Anywhere in the DNA strand, they can make a cut. And in that, these endonucleases, of course, there are generalized endonucleases which do not have any specificity with regard to their sites where they make the cuts. The other ones, they have restricted activities, like they are very specialized, and uh, which was, shown by scientists like Arbor, Nathans, and Hamilton Smith, who received the Nobel Prize in 1978 for their work. They showed that among the endonucleases, certain endonucleases, they are very specific in making the cuts. So they call that activity restriction, restricted activity, restriction enzymes. Further, of course, many of their colleagues, when they looked into these particular parameters, they studied that there are, like different, they are very specific to the sequence, the thing, where they have to make the cuts. Someone in a short stretch, in that particular thing, in a pattern, they make a cut in the middle. Someone make a cut little away from that particular pattern where these patterns are found. They make cut recognition sites little away, or the things. For example, they have given eco R1, hint 3, and BAM H1. BAM H1. So of course, here the things are named, eco R1, Escherichia coli, strain R. So Haemophilus influenzae, next H1, Hind, and that the strain name. Bacillus amyloliquefaciens, the next column. BAM H1. So they, of course, there are restriction sites where they make the cuts. For example, GAA-TTC, in E. coli, whereas Hint3, AA-GCTT, whereas in BAM H1, they make cut at GGA-TGC. And the enzymes, of course, once they were discovered, people tried, started discovering, looking into such kind of enzymes for different sources. Majority of them, majority of them come from bacteria. For example, eco R1, Escherichia coli, eco R1, R2, Escherichia coli, BAM H1, Bacillus amyloliquefaciens, Hint3, Haemophilus influenzae, then TAC1. Of course, here at this point I want to make it clear. Thermus aquaticus. Aquaticus, from this, this TAC1, which was derived, it is also made use in PCR, So this TAC, which is derived from Thermus aquaticus, which is a thermophilic organism which can tolerate boiling temperature, whereas the normal, the DNA enzyme, DNA polymerase, It gets denaturated. That's why it can't be used. Actually, the discovery of TAC H1, thermos from the thing. So actually, thermos aquatic was isolated by T.D. Brock from the Yellowstone Park, uh, you know, the things near New York City, Yellowstone National Park. So that is a hot spring, sulfur hot spring thing. So they're isolated. From that, did he, derive this enzyme? Then not one, Nocardia otitidus. Then Hinefan, Haemophilus influenzae. Like that, you have, SAW 3A1, Staphylococcus aureus, PVO2, Proteus vulgaris, then small one, Serratia marsusans, A3, Haemophilus Egypticus. Then you have HGA1, Haemophilus gallinarum, Olu 1, Orthobacter luteus, Capian 1, Klebsiella pneumoniae, Aurelia 1, Rhodopseudomonas, it is the Rhodopseudomonas spheroidus. Then Clavan, so Corine Phantom Latrum. So all that, of course, like that, if you can go on for a very long list of enzymes derived from bacteria. And these restriction enzymes, they may make two types of cuts. That is, the one on the top, left hand side, you can see, they make staggered cut. That is, you call sticky ends. You must have seen in your native the carpenters, while bringing together two logs, what they do, they cut two staggered cuts, to make a peg in that. If it is the blunt end cut, like down, if you say, it becomes a weak point, blunt end cut, as in hay3. Of course, the enzymes which are widely used in restriction, genetic engineering, transgenic studies, they are the one, which makes sticky ends or staggered cuts. And of course, sometimes they are also used blunt end cut enzymes which make blunt end cuts. But here, of course, certain enzymatic processing is needed to make it more stronger. And of course, the the examples which are given for different things, all of them, staggered end cuts, which makes staggered end restriction cleavage sites. And of course, in this slide, you can see the enzymes which make blunt end cuts and staggered end cuts. And cuts, of course, blunt end cuts, you can't underestimate. They are also made use for specific purposes by molecular biologists. In a sticky end, your foreign molecule, DNA molecule, while it should also have the same similar cut. That is in staggered end cuts. Whereas in blunt end cuts, it's not a matter. the middle, a piece of the DNA which also carry, has the blunt end, and it can be, of course, cemented by specific enzymes. Enzymes, again, similar examples how they make cuts. Alu, then H3, and Hind 2, blunt end cutters, whereas others staggered end cutters. Then you have enzymes, of course, these restriction enzymes. There are two kinds, isosisomers and neosisomers. What are they? Based on their behavior. Isosisomers are the restriction enzymes that have the same recognition site as well as the same cleavage sites. They are called as isosisomers. Means the recognition sites, within that recognition sites, they'll be able to make a cut, not away. Whereas in neosisomers, restriction enzymes that have the same recognition sequence but cleavage the DNA at a different site within that sequence, like for example, they have given SMA 1, XMA 1, and so on, like, of course, for them to act, they need that restriction site. But while cutting, they make a cut slightly away from that restriction site. Just the things. Many times it comes to, it is of very, novel help. So all that the molecular biologists have made use. And these restriction enzymes, there are three types of restriction enzymes that are used. The one, type 1. So the restriction enzymes, cleavage site, random, around 1000 base pairs away from the recognition site. Location of the methylase. Endonuclease and methylase located on a single protein molecule. And methylase located in a single protein molecule. Okay, in the same restriction enzyme. Then examples echo K1, echo A1, CFR1. Type 2, specific within the recognition site. They are the endonucleases. Endonuclease and methylase are separate entities. Echo R1, BAM H1, and Hint3, they are the examples, which are actually. Then type 3, they are random in their action, 24 to 26 base pairs away from the recognition site. Then endonuclease and methylase located on a single protein molecule. For example, echo P1, Hin F3, echo P15-1. So, among these three types of the restriction enzymes, to consider the one which are in the middle, type 2 restriction enzymes are of immense use, widely used, while molecular biologists in genetic engineering. So the type 2 restriction enzymes, of course, you can check for them, you will get large number of examples. So the endonuclease, it is within the, it happens, restrict, the what you call cleavage happens within the restriction site, within the recognition site. So that's the thing, advantage. It saves time for the molecular biologists. And of course, later, of course, the other thing that is required, methylation of the DNA that is transferred. So this is important because by methylation, the bacterial nucleases which are there in the cytoplasm, the bacteria which receives recipient bacterium will not be able to cause the damage to the nuclear material, recombinant, material that is transferred, the vector carrying the foreign DNA. So, of course, all that's given here for you. Applications of restriction enzymes. Of course, it is also used other than the genetic engineering, mainly by plant, breeders in mapping, gene mapping. They do earlier. They were doing it traditionally by uh, cytological techniques. They were using, but now it has become easy after the discovery of these restriction enzymes, restriction mapping. Then the recombinant DNA technology, of course, they are mainly this technique, recombinant DNA technology or genetic engineering or transgenic technique, one and the same. Everything depends on two things. The backbone for this, one is the vector molecules, the other one is these restriction enzymes. The vector molecules, the vector molecules, uh, the vector molecules, uh, yeah, like they are very important, which are actually to which you put clone your DNA piece of interest, a gene of interest, foreign gene, and make it a recombinant vector. For example, recombinant plasmid. Um, Here, of course, two kinds. If your interest is only amplification of the very traces of DNA uh, that is available to amplify it and get the like visible amounts of DNA. But you should know visible means not in human eyes. Then in the molecular biology techniques, in picograms to micrograms, at the most you can derive. So that particular thing, cloning. The second one, if your interest is to see that the recombinant product, then further you make use of another subsequent type of vectors, that is the expression vectors, which goes up to translation, the product. In this process, of course, you should remember the works of the first work, Southern blotting technique by Edwin Southern, that is DNA hybridization. Second one, the other works, later, the northern blotting technique. It is the RNA DNA hybridization, that is RNA, that is mRNA, that is extracted. The third technique, it is mainly based on immune reaction, antigen antibody reaction, where your expressed end product, when it is injected into the experimental animals like mice or rabbits, collect the blood and derive antisera and see whether that activity is there. And of course, this is shown in another technique, blotting technique called Southern blotting techniques. Kindly go through online; it's available. Southern, Northern, and Western. Sorry, last one is Western blotting technique. So all of them with the scientist name. Next, you have restriction enzymes Of course, cloning it is done, then single nucleotide polymerase, then restriction fragment length polymorphism in that technique, or AFLP technique also. DNA fingerprinting, of course, it is widely used. These things. So with that, you know, we have just just shown. I try to show you the things. Some little information on these restriction enzymes. Hmm. So now we will move to last one, which is part of this microbial genetics, molecular basis of mutation or mutation simply. Of course, mutation, it depends on the thing. While discussing on that, you need the molecular things, how it happens. Then what is mutation? It is a sudden heritable change in the characters of organisms. It may arise due to change in the base sequence of a gene mutation. Gene, you call it as gene mutation. It may result in change in chromosomal number and the structure, you call it as chromosomal mutation. So the gene mutation and chromosomal mutation. And of course, history, you'll get, in our biology, have studied a lot of things about this. Um, the earliest records of this mutation dates back to 1791 when an English farmer, yes, right, discovered a male lamb with usually short legs. The term mutation was introduced in 1900 by Hugo de Vries. The mutation, of course, the words that was heritable change, sudden heritable change that is introduced into an organism. Then systematic study on mutation began in 1910 with the discovery of genetic analysis of the white eye mutant Drosophila. So that is the thing, studies of Morgan. So he was actually using the things, Drosophila for his studies. Then Hetty Muller discovered mutagenic action of X-ray in 1927 in Drosophila. He awarded Nobel Prize for Physiology and Medicine in 1946. In 1929, Stadler described the mutagenic effects of X-rays in Borley. What are all the characteristics of mutation? Mutations are random events in terms of their occurrence and the gene in which they occur. It's very random, chance factor. So the duration taken or the place where it happened, all that, it's a sudden change. Most are harmful, but some are beneficial. Mutations may be recurrent. So, many times it may happen. Spontaneous mutations occur at low frequencies in nature. Of course, you should remember the evolutionary biologists, who have, of course, the phylogenetic tree given by, who is the scientist, phylogenetic tree, Yaro, universal, phylogeny. In that tree, of course, the ancestor is one. From that, it is three domain classification. The one side, the bacteria, the other side, archaea, the other side, the eukarya. And in eukarya, you know, so they further branch out into different eukaryotic organisms in the way to present existence. Of course, the humans or plants. Of course, plants are more and like ancient plants. Then, of course, various other organisms, eukaryotes, algae, then fungi and protozoa. They are all part of it. Eukaryotes. Then, among the higher organisms, you have the plants, then the animals. Of course, the insects, everything is covered here. So, the spontaneous mutations occur at low frequencies in nature. This is important. Just now, I told you from the single ancestor how different organisms of present day existence evolved. That evolution was made possible only by mutation. Mutation is one of the important causes for that, causes for evolutionary changes. Okay. Rate of spontaneous mutation varies from one to other gene. Uh, then some genes show high rates of spontaneous mutations. Of course, they are known to be more little susceptible to that mutation, known as mutable genes. The last ones, some other suppress the mutation rate of other gene called anti-mutator genes. Of course, one which introduces change. There is also, of course, a DNA repair mechanism in the living organisms. From the other end, they try to avoid this, the suppressing the mutations, ill effects of mutation. That is, with the help of the molecules anti-mutator genes. Mutation producing same phenotype effect may be located at different sites within the same gene. Then some sites within the gene show high rates of mutation. Such sites are called as hotspots. Of course, I've heard about it, more oftenly in our biology, PUC biology, hotspots, which are, where the mutation can occur at a very high rate. It can occur in any tissue or cell of an organism, and mutational events can occur in any development stages of an organism. Of course, it need at a particular, not well defined. It may occur at any stage of an organism's life cycle. Mutations occur in both forward and reverse directions. This is important. Mutation, that change that is caused because you have two strands. Uh, the the thing that is the changes that is introduced need not be in the 5 prime 3 prime in direction. It may be from the reverse direction also. The direction also. Then rate of forward mutations are higher than those reverse mutations, of course. However, among these two, the forward mutations are more frequent. Most mutant alleles are pleiotropic. Then, just little information on the mutagens. So the mutagens capable of, the molecules which are capable of introducing mutations are called mutagens. Mutagens, they are agents, the molecules which can introduce. They are classified into two types: physical mutagens and chemical mutagens. Physical mutagens, of course, we have studied. UV rays are also dangerous. X-rays are also dangerous. Gamma rays are highly dangerous, but in cancer treatment, you can't avoid that, because the at site treatment, gamma rays are made use. You can't avoid them. But then, of course, scanning, X-rays, CAT scan, PET scan, then MRI scan, and all. The advanced scanning techniques, they are more dangerous.
Okay, okay.
Several times, several, several means, so several hundred times, more dangerous than the simple X-rays, 400, 500 times. But however, for treatment, you can't avoid them. The one, all these innovations, they also carry,
Yeah,
they
usually
they
even
also carry the
there is no need of asking them.
uh,
Just what
dangers also.
to them.
Then chemical mutagens, a large number of chemicals like insecticides, of course, many of them are carcinogenics, fungicides, herbicides. Of course, you are aware of the, uh, in the thing, initial days of
Yeah,
Green
yeah.
Revolution around
This all thing
1965,
coming
when
from our URL, Prasad
because of the
Prasad,
shortage of foods, when
it is
new
from our
varieties were
childhood, right?
introduced,
Yeah,
then
while we are giving the vendor money,
high yielding varieties were
only
brought into India,
India.
of course, ignoring the
Ignoring
native varieties because
without any use,
which produce
nobody asked for that.
large amount of hay compared to the
Just give,
rice
just asking that.
and then
Okay, okay. Keep on.
they started because to grow them they were highly susceptible for weeds, diseases, insect pests, all that, all these innovations, recent things, so-called you use the word pesticides, which includes weedicides, insecticides, fungicides, all of them, they were introduced, of course, uh, which actually in the initial days the danger of that was not visible, but slowly the thing, frequency of cancer, especially the Western Ghats and the coastal regions, where people live in between nature, who are not exposed to the all these chemicals, in those belts, sudden appearance of cancer was reported, and of course nowadays it has become rampant. That's one aspect. That's why the present day, of course, we are like we want to go behind organic farming. Of course, it's too late now. No specific thing, attempts, specialized attempts has been done in that direction. Then, of course, the one, which are many chemicals which are used in in molecular biology, like ethidium bromide. In DNA gel electrophoresis, ethidium bromide is used. Then acriflavin is used. Acridine orange is used. Like that, you have several things. They are again, you know, mutagenic. The people who are using have to be, I think, very careful. And another thing, you should remember the actively multiplying cells, especially adolescents and youths, where there is the thing, so the sudden, growth occurs, So the increased growth occurs in them. They are more susceptible. They have to be very careful as compared to the people who are aged, because the cell multiplication rate is decreased. Then the causes of mutation, errors in DNA replication, uh, on a very rare occasion, DNA polymerase will incorporate a non-complementary base into daughter strand. During the next round of replication, the misincorporated base would lead to a mutation. This, however, is very rare as the exonuclease functions as a foolproof, proofreading mechanism, recognizing mismatched pairs and excise them, remove them. Second possible things, errors in DNA recombinations. DNA often rearranges itself by a process called recombination, which proceeds through a variety of mechanisms. Occasionally, DNA is lost during replication, leading to a mutation. Then the chemical mutagens, many chemical mutagens, of course, some exogenous, man-made things, of course, all these, these pesticides, recent, things, which you call uh, xenobiotic chemicals. I think, many of them are cancer causing. Many chemotherapeutic drugs and intercalating agent drugs function by damaging DNA. So that's the thing. Then radiation is, of course, X-ray, then UV rays. While handling, for example, laminar airflow system, you have UV rays. They're used to sterilize the thing in a laminar airflow cabin, but you should be very careful. Unless it is put off, then you should not, put your hands there. X-rays, of course. Don't attempt for more often X-rays. As far as possible, you try to avoid. Then gamma rays, of course. Gamma rays. It is only certain treatments. That is Mobile 60. That is advised for treating cancers, particular type of localized cancers, and you have to think and use before taking any decisions. Then types of mutation based on cytological basis, gene mutation and chromosomal mutation, based on effect on phenotype or tissue or of origin, you classify them into somatic mutation and genetic mutation. The genetic mutation is also called as germline mutation. Based on causes of their origin, spontaneous mutation, induced mutation, types of traits affected, visible and biochemical mutations. So, based on the effect of tissue of origin, somatic mutations arise in the somatic cells, passed on to the cells through the process of mitosis. Effect of these mutations depends on the type of the cells in which they occur. For example, in a living organism, certain cells, may not be directly involved in the activities of an organism, like plants or humans or any other thing. So, in that case, it may go unnoticed. In case if it is associated with the affected thing, it is present in a important site, then it's going to bother us most. Effect of these mutations depends on the type of the cell in which they occur and developmental stage of the organism. So at different stages of an organism, it occurs early in development, larger the clone of the mutated cells. Gene mutations, they occur in the cells that produce gametes. They are also called as, just now I had told you, germline mutations, passed on to future generations, because it is through the gametes it happens. So in multicellular organisms, the term mutation is generally used for germline mutations. Some common types, the ones which are found deletions means, like during this mutation, introducing mutation, a part of the gene is removed. Then inversion, all that we will discuss now. In the future, it is a when a part of the chromosome is left out, then it is deletion. The when part of the chromatid breaks off and attaches to its sister chromatid insertion occurs. The result is a duplication of genes on the same chromosome. Means the same information is present, which may not be required. Then third type, when part of a chromosome breaks off and attaches backwards, it is inversion mutation. It is inversion mutation. So when a part of the chromosome breaks off and reattaches backwards, here it is shown, reverse direction. It is reverse, I think, it is inversion. When part of one chromosome breaks off and is added to a different chromosome, a translocation occurs. Translocation. You know, site changes. Translocation. The site changes. So when part of one chromosome breaks off and is added to a different chromosome, a translocation occurs. Due to breakage, a piece of a chromosome, all that, how it happens. They have pictorially shown this inversion. So the deletion, inversion, duplication, then translocation, all that. Gene mutations, of course, they are very important. They are the one, because so the offsprings, are affected. Point mutation, missense mutation, nonsense mutation, frame shift mutation, all that. So we will stop at this stage. We will continue in the tomorrow's class. Okay.
Okay, sir.
Uh,
Okay, sir. Thank you, sir.
Tomorrow between 2:15 to 3:15.
Yes, sir.
Okay,
Okay.
sir.
Okay.
Thank you, sir.
Thank you. So bye. Take care of the health, of your health. Don't go outside. Hmm. Register for
Okay, sir.
vaccination. Huh?
Yes, yes, sir.
You know, keep yourself warm. So okay. Bye.
Thank you, sir.
Thank you. Bye.
Bye.
Bye.